Adenine is a purine with a double ring structure. In double-stranded DNA, adenine base pairs with thymine. Guanine is a purine with a double ring structure. In double-stranded DNA, guanine base pairs with cytosine. Thymine is a pyrimidine with a single ring structure. In double-stranded DNA, thymine base pairs with adenine. Cytosine is a pyrimidine with a single ring structure. In double-stranded DNA, cytosine base pairs with guanine.
DNA exists as a double helix, with the bases of the two DNA strands linked to each other by hydrogen bonds. For DNA to replicate, three events must occur: strand separation, complementary base pairing, and nucleotide joining. During strand separation, an enzyme called DNA helicase uncoils and separates the double helix. DNA helicase separates the two DNA strands by breaking the hydrogen bonds connecting the base pairs on the two strands. In the murder investigation on Prince Edward Island, Marilyn Minotti Raymond and her colleagues used heat rather than enzymes to break the hydrogen bonds and separate the strands of Snowball's DNA. In complementary base pairing, the unpaired bases on each of the DNA strands form hydrogen bonds with the complementary bases of free nucleotides that have diffused into the area. Regardless of whether the base is on the parent strand or free nucleotides, adenine binds only with thymine, and guanine binds only with cytosine. In nucleotide joining or polymerization, the enzyme DNA polymerase links the nucleotides together in the new DNA strands. Note how both of the new DNA strands are formed in the 5' to 3' direction. Because of complementary base pairing, each double-stranded daughter DNA molecule is identical to the parent DNA molecule. Note that each of the two daughter DNA molecules has one strand intact from the parent DNA molecule and one completely new strand. Because only one of the two strands in the daughter DNA molecules is conserved from the parent DNA molecule, this type of replication is called semi-conservative replication.